Hello everyone. This week in lab we're going to talk about biochemical tests that would help us identify and differentiate between pathogenic Cocci bacteria. Now we're going to be able to identify these organisms based on what they can metabolize. For example, whether they're capable of metabolizing certain sugars or not, or whether they're able to metabolize certain proteins or not. We're also going to be able to differentiate between them and identify them based on what type of enzymes they can make and produce. Now, this PowerPoint is specifically going to we're going to focus on gram-positive bacteria, and we're going to focus on those that usually are medically important because they can cause like problems because some of them are pathogenic. So that's why we are going to go ahead and discuss them since, like, these, as I said, bacteria can cause some medical problems and they can be pathogenic. So the genre that we're going to talk about is Staphylococcus. Now, Staphylococcus is the genus. Now, there are multiple species of Staphylococcus out there. So we're going to talk about Staphylococcus in general. Now, Staphylococcus is usually found on skin, or it could actually be found in the nose, okay? Even if you're healthy and everything, there could be some Staphylococcus in the nose. Now, not all Staphylococcus are pathogenic, but some of them are, okay? We're also going to talk about Streptococcus. Now, again, Streptococcus, that's another genus. Now, there are multiple Streptococci species out there. Again, not all of them are pathogenic, but some of them are. And Streptococcus it could be found in the upper respiratory tract. And Streptococcus bacteria, one thing to note about them is they like to stay in conditions that have a bit of like lower oxygen levels, okay? So that's kind of like an environment they prefer, okay, like lower oxygen levels. The first test that we're going to talk about, it's a broth test. So it's usually done in a test tube where we have a broth. And so this first test that we're going to talk about is called the Litmus Milk test. Now, this is how a litmus milk test would look like, and this is actually a test tube that's uninoculated, meaning it doesn't have any bacteria in it, okay? It's uninoculated, and it comes in this purplish color, okay? It's kind of like a purple milky broth. Now, this broth contains milk. Okay, so that's why we call it milk test. It actually contains milk, and it contains something called litmus, and litmus is a pH indicator. We've talked about pH indicators before, but what is a pH indicator? It's basically a chemical that's going to change color based on the condition of the media. So if the media is neutral, basic, or acidic, that litmus color is going to change its color. So purple is actually the color of neutral, okay? Like a neutral solution, it would actually be purplish in color. So let's go ahead and take a look at, what the litmus milk tests for, and let's take a look at some of the results together. So litmus milk test, one more time, it contains a complex mix of carbohydrates. What type of carbohydrates? Well, mostly lactose. Why lactose? Because, as I said, this test has milk, and milk has sugar. Now, what type of sugar is in milk? Well, it's lactose, okay? So there's going to be a bunch of lactose in this media. And then milk also contains proteins. And now, what type of protein does milk contain? Mostly casein. Now, there are, of course, like other proteins, but mostly casein. So there are two important, nutrients in this broth. There is the lactose sugar, and there's the casein protein. Now, what we want to do with this test is we want to figure out and differentiate, right, between our bacteria. We want to be able to differentiate between the bacteria that are going to be capable of metabolizing lactose and actually fermenting it. And we want to be able to also determine, whether the bacteria we're testing is capable of metabolizing the protein, metabolizing the casein. Now, one thing to note is it's going to be one or the other, okay? So either the bacteria are going to ferment the sugar or the bacteria are going to metabolize the protein. It's not going to be both, okay? So the bacteria is going to just do one or the other. And one more thing is also in this broth, as I said, we add litmus. And litmus is a pH indicator that's going to change color based on, the acidity or alkalinity of the broth. Okay. Now, we start off with an uninoculated test tube, okay? And as I said, this uninoculated broth has the milk and has the litmus in it, and it starts out kind of like a purple broth, and this is like the neutral color, okay? So it starts out purple. Now, after we go ahead and we inoculate this tube, and we usually inoculate it using an inoculating loop. So you would, again, you would follow the aseptic technique of how to use the loop and how to kind of like pick a sample of bacteria. And then you would inoculate this test tube, and then you would go ahead and put it in the incubator. Now, what are some results that you would get? Well, one of the results that you would get is the broth goes from that purplish color to that pink color. Okay, now if your broth turns pink, what does that mean? Well, it means that your bacteria was actually capable of fermenting the sugar. Which sugar are we talking about? Lactose. So it was capable of fermenting the sugar and it was capable of producing an acid, okay, an acidic product when it was fermenting the sugar, turning the broth acidic and turning it into that pink color. Now, one thing to note about the litmus milk test is that it can actually take a while, and over time, as time progresses, your results might change. So after you inoculate, right, like this test tube right here, and you put the test tube in the incubator, and then we usually leave it for like 24 to 48 hours. Now, within 24 to 48 hours, you might see something like this, that your broth has turned pink, which means that your bacteria, is fermenting the lactose sugar. But what we usually do is we go ahead and we take this test tube right here, this pink one, and we actually stick it back in the incubator. So you stick it back in the incubator, for a few more days, just to kind of see, okay, if anything is going to change. Now, let's say you did that, right? You took this test tube and you incubated it for an extra, okay, a couple or, 3 days. What you could see is something like this. Now, it's still pink, right? But notice how it developed that white clot. It's kind of like a clump, okay? It's going to look, there's going to be a clumpy, white thing at the bottom of the tube. And when you flicker the tube, it's not going to dissolve, okay? So that white clump is going to be at the bottom of the tube and it's going to be solid. Now, what is that white clump? This is actually the milk curdling. And basically this is curd, okay? So this is curd forming. Now, what's curd? Okay. If you've ever, taken a batch of milk that you have at home and, give it a try. So get some milk, okay? Put some milk in a pot and then go ahead and add some vinegar to that milk or add lemon juice to that milk and watch what's going to happen. So you're going to notice that when you add something acidic like lemon juice or vinegar to your milk, um, and you can kind of like speed the process up by actually kind of like heating your milk, okay? You're going to notice that your milk is going to start to curdle. It's going to start kind of, becoming clumpy, okay? And this is basically what's happening right there. Now, if this happens and you get this curd at the bottom of the test tube, what does that mean about our bacteria? It actually means that your bacteria is going through more sugar fermentation, like extreme sugar fermentation. And as I said, with more fermentation, more acid is going to be produced. And therefore, because there's more acid being produced, the milk is going to curdle, right? Kind of like how I just explained, putting something acidic to your milk is going to start to curdle. And basically, this is what the bacteria is doing right now. It's producing a lot of acid because it's fermenting the lactose sugar, causing the milk to curdle. So this kind of like a result right here, as I said, this is after you've incubated this test tube for a bit longer. So as I said, like, it could take up to 7 days for your results to develop. So within, as I said, like the first day or two, you will see some change, and then if you leave it longer in the incubator, you might actually see more change. Now, does that mean I must get curd if I left it longer in the incubator? No, that's not always the case. Again, at the end of the day, it depends on what type of bacteria you have. Some bacteria are just going to go through sugar fermentation and that's it. So even if you incubate this test tube right here for a longer period of time, they're still not going to go through extreme sugar fermentation. Okay, but some bacteria strains, if you go ahead and leave the test tube longer in the incubator, it is going to go through more sugar fermentation and you're going to see that curd at the bottom of the test tube. Okay. So this is, this is a group right here of some results that you can get, okay? And so if you get, as I said, like this pink color, okay, and you basically went this pathway, or the bacteria went this pathway, then your bacteria is basically fermenting the sugar, okay? Now, but there is also another pathway, okay, that your bacteria could take, and it could basically go this pathway, okay, and you would get this result right here. So as I said, it's either or, okay? Your bacteria can't go through both pathways. It's going to choose, okay? I mean, not that it's choosing, okay? This is what the bacteria is gonna, meant to do. It's either, going to ferment the sugar, or it's going to go ahead and go through the protein breakdown. And by the way, it's not like your bacteria is going to choose, today I'm going to choose to ferment the sugar, but tomorrow I'm going to choose to break down the protein. It doesn't work that way, okay? It's one pathway or the other in that pathway is like set, okay, for that bacteria. So what is, what's the, what could be another pathway that you could see with, with some bacteria? Well, some bacteria out there cannot ferment sugar, but what they can do is they can break down and metabolize the proteins. Now, where's that protein coming from? Remember, milk has both the sugar and it has the protein, okay? So some bacteria out there, even though they can't ferment the lactose, what they're capable of doing is they are capable of metabolizing the casein, and that's then what they will do, right? And so therefore, they're going to go down this pathway, right? And so what they're going to do is metabolize the protein. How am I going to be able to tell whether they're metabolizing the protein or not? The color of the broth is going to turn like a dark purplish, bluish broth. Okay, so if it turns like dark purple or blue, that means the protein is being broken down. Now, as I said, this could be the result that you would get after like 24, 48 hours of incubating your broth. Now, what could happen, if you go ahead and incubate this broth longer? Well, if you incubate the broth longer, then you can go through these results over here, and that's where your bacteria is basically, let's take a look at that first one right here. So this one right here, the broth is still purple, right? But you're getting some like white precipitate at the bottom. Now this white precipitate at the bottom is not curd. So is this white precipitate right here the same as this curd that we were talking about? No, it's not. And one way for you not to confuse it with the curd, remember I said the curd is solid. So when you go ahead, kind of like flicker your tube, kind of shake it, that curd is going to be solid, okay? Now, this white precipitate, if you actually like flicker your test tube, it might actually dissolve, okay? So it's not solid as the curd. Okay, what is that white precipitate? So the white precipitate is actually the litmus, right? That pH indicator that we added in here. That litmus is actually being reduced. What is reduced? Reduced meaning it's going through the redox reaction. And we talked about reduction before. What is reduction? Remember the mnemonic oil rig? So reduction is gain. So if the litmus chemical, that pH chemical, is actually gaining electrons, it's going to be going through reduction, and as it's gaining electrons, basically this litmus is going to kind of give you that white precipitate, and it's going to precipitate at the bottom of the test tube. Okay. Now, if, as I said, like, through after you've incubated it even longer, as I said, I mean, it could take 7 days, right? So throughout those 7 days, the results could keep changing. Now, what could happen is the broth could turn brown, okay? Or sometimes it's not completely brown, okay? And sometimes like the color is just kind of changing, the bottom or like at the top. And sometimes it's not really brown, but it's kind of like your milk is now turning clear. Now, if you think about it, milk is not translucent, right? You guys, meaning that it's kind of like opaque, right? And the reason why it's opaque because of all of that protein milk has, okay? Now, what's happening here, right? What if the bacteria is going down this pathway? What is the bacteria doing to that protein in milk? Well, it's breaking it down, remember? So if the bacteria continues to break down that protein further and further, what's going to happen is the milk is not going to become opaque anymore because the protein in the milk is being broken down. And this is kind of what's happening right here. That's why, the broth is going to start becoming, that kind of clear. It's going to sometimes, as I said, like not completely brown, but it's going to have like that straw, okay, like that straw color, like, straw hay color to it. And we call that process now the protein is being completely broken down. We call that process peptinization. So peptinization is the process where the protein is completely being broken down. Okay. And so, as I said, your bacteria, okay, that you are testing, it's either going to choose to go down this pathway right? Ferment the sugar, or it's going to go down this pathway. And again, this depends on the bacteria strain. Some bacteria are capable of fermenting sugar, and therefore they won't metabolize the protein. And then some bacteria, they're going to metabolize the protein, and therefore they're not going to ferment the sugar. So these are some results, okay, of what you could get if you do the litmus milk test. Now, those are, some more images for you, kind of actual images so that you guys can kind of see what's, how it really looks like. So again, this is kind of how you start it off, okay? You start off with an uninoculated broth. And as I said, it contains milk, and that's why it's kind of opaque, right? Because of all of that protein milk has, the casein protein. But again, what milk contains as well is the lactose, okay? So there's definitely the protein casein in here. There's the lactose sugar. What else is in this test tube? The litmus, right? The pH indicator, litmus, okay? After you go ahead and inoculate the test tube, let's say you get this kind of dark blue, okay, kind of purplish color. So what does that mean? It means that your bacteria in here is capable of breaking down the protein, right? And that means that now the condition in here is becoming basic, it's becoming alkaline, okay? And the color is going to turn that bluish color that you see, okay? Now, what if the solution, as you can see, is kind of, blue, but now you have that white precipitation at the bottom? Now, that means, remember, that you are, you're getting the litmus reduction, right? So when you see that white precipitation, that means litmus reduction is taking place. And the protein is, is being broken down, okay? And also when you see this kind of like, litmus reduction at the bottom, this is kind of telling you that your bacteria is actually starting to break down the protein even further, and peptinization, is also kind of like, an indicative that peptinization is, is kind of taking place, right? But for you to kind of say that, okay, I have complete breakdown of the protein and the peptinization is, peptinization definitely took place and it's complete. As I said, you get this kind of like straw color, right? And that means, and see how kind of translucent it is compared to how opaque we started off with. And the reason why it's translucent now, because the protein now is completely broken down, okay? Okay, so that's in case the bacteria was breaking down the protein. But what if the bacteria is not breaking down the protein, but it's fermenting the sugar? Then what are we going to get? Well, you're going to get, for example, this like pink color right here, and that means that the sugar is being fermented, and therefore acid is being produced, and the condition now is acidic in here. And if the test tube was left longer in the incubator, then the bacteria could, as I said, it's not a must, okay, but the bacteria could go ahead and ferment the sugar even further, and that's when you get the curdle, right? That's when you get that curd, right? That because the milk is now curdling, and that would indicate that there's extreme sugar fermentation taking place, and that's why you're seeing that white curd, and as I said, it's like a solid white curd, okay? So those are, some results for the litmus milk test that you might get. Now there's another test that we're going to talk about, and it's another test done in a test tube, okay? So it's another broth test, and so you start off with, with some broth. Now this test right here, it's called the nitrate reduction test. And we start off, this is how it looks like when the broth is uninoculated. It kind of has that clear yellowish color to it. Now it's called the nitrate reduction test because we add in this broth, we add nitrate. Okay, so that's why it's called the nitrate reduction test, because you add in here some nitrate. Okay, now what is this test used for? Well, this test, as I said, first of all, you start off with a broth that has nitrate. And this test, we want to know whether your bacteria has the capability of reducing nitrate. Again, what does reducing mean? Reducing doesn't mean breakdown, okay, guys? So reducing means we want to know whether your bacteria is capable of reducing the nitrate, meaning whether nitrate is capable of accepting electrons, right? Remember the redox reaction one more time. Oil rig reduction is gain. So we want to know whether the nitrate is going to accept electrons. Okay, now if you recall, okay, if you go back to the PowerPoint where we were talking about aerobic versus anaerobic respiration for our bacteria, if you recall, we said that bacteria that are capable of going through anaerobic respiration, those bacteria can actually use other things like nitrate, right, instead of oxygen as their final electron acceptor during the electron transport chain. So one more time, we said that bacteria that are capable of going through anaerobic respiration, they're capable of using nitrate as their final electron acceptor during the electron transport chain. And so what does that mean? Nitrate being the final electron acceptor, meaning that those bacteria are actually going to use the nitrate, okay, so that this nitrate, okay, so that the nitrate in their media or in their environment can go ahead and accept all of these electrons during the electron transport chain. And that's what this test is going to tell us. The test is going to tell us whether the bacteria, is capable of, of doing that, yes or no, okay? And so we want to know whether nitrate is being reduced. One more time, when I say we want to know whether nitrate is being reduced, that doesn't mean broken down. It means whether it's going to gain those electrons or not. So this is how nitrate looks like, right? This is like the compound. NO3 is nitrate. Now, if nitrate is being reduced and it does accept and gain electrons, then it might go ahead and, not might, this is what happens. So when nitrate goes ahead and gains electrons, it turns into nitrite. So this one is nitrite and it's NO2. Now, when nitrite gains electrons, it's going to go ahead and turn into NO. What is NO? NO is nitric oxide. Now, if nitric oxide goes through reduction, then you can get nitrogen gas. Now, does that mean all bacteria that are capable of reducing nitrate have to go through this whole entire process? Absolutely not. Some bacteria, as their nitrate, as the nitrate is being reduced, it will only be reduced to nitrite and that's it. Okay, so the reaction stops right here. But then some bacteria out there have the capability of going all the way to the nitrogen gas. Okay, so one more time, it depends on the bacteria strain that we are talking about. Some of them can go all the way, so let me kind of draw an arrow. So some of them can actually go all the way to nitrogen gas. Okay, so they would go through, you can kind of call it like they're going to go through a full reduction. Okay, so the nitrate is going to go through a full reduction and go to the nitrogen gas. But then sometimes, the nitrate is just going to go and get reduced to nitrite, and that's it. Okay. So, as I said, it just depends on the bacteria strain that you are talking about. And so, um, if nitrate only goes to nitrite, then we usually call this a partial reduction, okay? But if it goes all the way to nitrogen gas, we call that a full reduction. And as I said, depending on the bacteria strain, one bacteria could go through the full reduction, and then one bacteria could go through the partial reduction, okay? So usually what we do, right, we get this broth, we go ahead, which contains nitrate, of course, right? Your broth has to contain nitrate. And then you go ahead and you inoculate the broth with your bacteria. You stick it in the incubator for a couple of days. Now, after you take it out of the incubator, you go ahead and you add a few reagents. So you add like 3 drops of a certain reagent. We're just going to call it reagent A. And then you add another 3 drops of another reagent, and we're just going to call it reagent B for now. Okay. And again, when do you add those 2 reagents? You add them after you've incubated your bacteria, right? After you've incubated your broth containing the bacteria, this is when you add those 2 reagents. Now, we're going to talk about the color changes that you get, and I'll show you images. But if, let's say, you added those 2 reagents and then nothing, you don't see anything, right? You still see that your broth is yellow and you don't see really any color change, then what you could do is add a pinch of powder zinc. Do be careful when you're handling powder zinc, because powder zinc is hazardous, okay? So you have to be very careful when you are handling powdered zinc, okay? And you can go ahead and kind of use a wooden applicator, like, take some zinc and then kind of sprinkle it in your broth and then see what would happen, okay? So we'll go ahead and take a look at the results together. Okay, so here's a table to kind of just to make it a bit easier for you, because the results for the nitrate reduction test could be a bit confusing, because one color could mean 2 things, okay? So one color that you get might mean 2 things, and it depends when did you get to see this color. So let's go ahead, kind of, show you to make more sense of what I'm saying. So let's say right here, here is your broth, okay? And let's say that this broth is, turbid, so it has bacteria growth. So this is like the broth that you took out of the incubator, okay? So what's the first thing you're going to do to that turbid broth? Well, you're going to add the 2 reagents, right? Remember, you're going to add the 2 reagents. Okay, so let's say you added the 2 reagents and your broth turned pink, or, that pinkish, like reddish color, okay? What does that mean? If it turns red, okay, but as I said, it's super important to kind of note when did you see that red color, because red in this test, you guys, could mean several things, okay? So that's why I'm saying make sure that you kind of know when did you see the red color. If you saw the red color right after adding those 2 reagents, well, that means that this is a positive result. It means that your nitrate was reduced to nitrite. That means the bacteria was capable of, going through that nitrate reduction, okay? It means basically your bacteria was capable of producing certain enzymes that did allow this whole entire thing to take place, the whole nitrate reduction to take place. So it's a positive result, okay, guys? But again, when do we consider pink to be positive? Right after the addition of those two reagents. Again, why am I stressing so much of, like, you know, the idea of when are you seeing this red color? Because you guys are going to see in a few minutes how red could mean something totally different. Okay, now let's say, okay, so this is kind of like, you know, one result that you would get. Okay, versus, let's say that one more time you started out with the turbid broth, right? After you've incubated it, and now you have bacteria growth, and you can see that you have growth because you see turbidity. Okay, let's say that you added the two reagents, okay, guys, and nothing happened. So you added the two reagents, and guess what? The broth is still yellow. Okay, what does that mean? Well, this means, okay, this is actually inconclusive. Okay, this is what it means. So if your results, after adding those two reagents was clear, this is inconclusive, meaning I am not sure, okay, whether the bacteria really didn't go through any reduction, or maybe it did go through reduction. So it's inconclusive. Okay, you're gonna go like, well, wait a second, it didn't, it's not pink. Well, yes, but remember how I said, let me kind of just write it out for you one more time. We said that some bacteria, right, can go through full reduction or they can go through partial reduction. So as I said, this can happen, right? Nitrate can be reduced to nitrite, which then can be reduced to nitric oxide, which then can be reduced to nitrogen gas. Okay, now if the nitrate was reduced to nitrite, okay, how are you going to be able to tell that this happened? Well, right here, right? If you've added, the two reagents and it turned pink right away, then we know that it went through a partial reduction and it went to, it was reduced to nitrite. But if it was reduced to nitric oxide or if it was reduced to nitrogen gas, guess what? You're going to get clear results after adding the two reagents. And that's why we say that our results are inconclusive, okay? Because it could mean that my bacteria did go through reduction, okay, but not just to nitrite. It actually went all the way to nitric oxide and nitrogen gas. Those two reagents cannot tell you that information. So therefore, what do we have to do if we get such inconclusive data? You have to go ahead and add zinc. So you would go ahead into this, broth, you would go ahead and add zinc. Now, let's see why we add zinc and let's see what type of, results you would get, okay? So let's say that after you've added the zinc, okay, guys, still nothing happened, okay? Let me actually, well, maybe it would be helpful actually to tell you what zinc does though, okay? So let me back it up and mention something about zinc, you guys, okay? Now, when you guys add zinc to nitrate, what's going to happen is zinc basically kind of acts as a catalyst, okay, guys? And zinc is going to help the reduction of nitrate to nitrite, okay? So, as I said, zinc is basically like a catalyst, so it's going to go ahead and help catalyze the reduction of nitrate to nitrite, okay? So, if you go ahead and add zinc, Okay, guys, so you went ahead and you added zinc. And you get a pink color, that would be negative. Okay, now, and that's why I was saying pink means two different things depending on when did you guys see the pink color. Now, if you see the pink color after adding zinc, okay guys, so after I've added zinc, I go ahead and I see a pink color, that means that my bacteria cannot reduce nitrate. Okay, let me explain why, because it's, kind of confusing. Now, if there was, okay, let me kind of write it right here. Okay, so in this broth, you guys, there is nitrate, right? Okay. Now, if after adding the two reagents, as I said, you still get this kind of yellow color, okay? Now, as I said, this is inconclusive data. Like, we're not very sure what's happening. We're not very sure whether the nitrate, okay, didn't get reduced at all, okay? Or it might have been reduced, but it got reduced to something like nitrogen oxide or nitrogen gas, okay? So what we do is we go ahead and add zinc. Now, what zinc is supposed to do is if there is nitrate in here, okay, so if there is nitrate in here, after you add the zinc, okay, after you add the zinc, it's supposed to go ahead and reduce it to nitrite. Okay, now if zinc goes ahead and reduces nitrate to nitrite, what's going to happen is it's going to change color and it's going to give you a red color. Okay, so you're going to get this pink color right here. Why are you getting pink color? Well, that means that there was nitrate. There was still some nitrate in the test tube. And because there was still nitrate in the test tube, zinc was able to go ahead and reduce it to nitrite. And that's why you get this pink color. But what it means, in terms of what does that mean in for my bacteria? Well, it means that your bacteria didn't do the reduction. Okay, it means that the nitrate that you started off with was still there, which tells you that your bacteria was not capable of reducing the nitrate. Okay, so the nitrate that you started off with was still in the broth. Okay, how can I prove that my nitrate was still in the broth and that means my bacteria did not reduce it? By adding zinc. Okay, if after you've added zinc, you get the pink color, that means that your bacteria was not capable of reducing nitrate. Okay, so do you guys see how important it is to kind of figure out when did you get this pink color? Because depending on where you get and you see this pink color, the pink color could mean something totally different. Okay, now if I've added zinc, okay, and I get clear, okay, I get a clear color. What does that mean? Well, it just means that zinc did not find any nitrate, okay, to reduce. Okay, so let's say that you've added zinc into this broth, okay, but there isn't any nitrate. Why isn't there any nitrate? Because the bacteria has already reduced it. Okay, so if the bacteria has already reduced the nitrate, if you add zinc, then zinc is not going to do anything because there isn't any nitrate for the zinc to reduce. Okay, so what does that mean? Well, it means that in this case, since there isn't any nitrate, it means that you probably have what in here? You probably have nitric oxide or nitrogen gas. Okay, so that's why in this case, clear would mean positive, because clear in this case means that your bacteria went through a reduction, and it reduced it either to nitric oxide or to nitrogen gas. Again, we're not going to be 100% sure whether it's a nitric oxide or nitrogen gas, but I mean, we can tell that it is definitely positive because there isn't any more nitrate in the test tube. So in this case, right, clear is positive, not negative. Okay, so clear over here after the addition of zinc is actually positive. Okay, so this is basically the nitrate reduction test. Okay. Okay. Okay, so you guys, one more time, depending on where you when did you see the pink color? If you see the pink color right after adding the first two reagents, that means that the bacteria was capable of reducing nitrate to nitrite, and so that would be positive. But if after adding the two reagents you get clear, well, you can't stop right here because you are still not sure, okay? What does that mean if it's clear? So if it's clear after adding the first two reagents, you must go ahead and add a pinch of zinc. Now, which test tubes would get the pinch of zinc? You would go ahead and add it to those that turned, clear in color. And so after adding the pinch of zinc, if it is pink, if the broth turns pink in color, well, that means that your bacteria did not reduce nitrate, because that would just mean that you still have nitrate in the broth. And so it means your bacteria was not able to reduce it. But if after adding zinc, again, it stayed clear in color, then in this case, it would mean positive, because it would mean that your bacteria did reduce the nitrate, maybe to nitric oxide or to nitrogen gas. So pink the first time would be positive for nitrate reduction. Pink the second time would be negative for nitrate reduction. And so one more time, after adding the two reagents, if it turns pink, then nitrate did get reduced to nitrite. If you've added zinc and you do get a pink color after adding the zinc, well, that means it's a negative result, and that would mean that your bacteria is not capable of reducing nitrate. Now, if you add zinc and you don't get any color change, well, that means it is a positive result and the nitrate was reduced all the way to either nitrogen oxide or, all the way to nitrogen gas. Now, some more tests to talk about. These are plate tests, so we usually do them like, on agar plates. This plate right here starts looking out like pink like this. This is an uninoculated plate, and we call it mannitol salt agar. And mannitol salt agar, it's called salt because there is a bunch of salt in here, and mannitol is actually a sugar. So mannitol is a type of sugar. So this plate has both sugar and salt, and of course some other stuff, but those are like the most two important ingredients, the sugar and the salt. We're also going to talk about another plate called the blood agar plate, and it's called blood because it actually has red blood cells. Not humans, but sheep red blood cells. Okay, and that's why it's kind of maroonish in color, because of course it has red blood cells. And then we're going to talk about a catalase test. Now, catalase is not the name of the media. So this media, could be a TSA plate, for example, a tryptic soy agar, but we use this plate to go ahead and perform a test called the catalase test. So I'll show you, how we do the catalase test in just a minute. But as I said, like this is a regular TSA plate. Okay, so this is a regular TSA. Now, does it have to always be TSA? No, you can use a regular nutrient agar plate also to do a catalase test. So not necessarily, okay? You don't always have to use a TSA for the catalase. Now, another thing to note about the blood agar and the TSA is we go ahead and we incubate them in a candle jar. A candle jar would be a capnophilic environment where the amount of oxygen is low and the amount of carbon dioxide is high. Now, since this PowerPoint we're talking about identifying and differentiating between Staphylococcus and Streptococcus bacteria. Now, Streptococcus as I said, they love to live in environments that don't have a whole lot of oxygen. So, Streptococcus, genre, they would actually prefer kind of like a capnophilic environment. So let's talk first about the mannitol salt agar. So as I said, this plate has two main ingredients. It has the sugar mannitol and it has salt. Okay, so this plate is definitely selective. Why is it selective? Because it has a bunch of salt in it, it's going to select for halophiles. What does that mean? It means that the halophiles are going to be the ones kind of like encouraged, right, to grow on it, to get cultured on it, because halophiles are the ones that love salt, right? And so therefore, if you have a bacteria that cannot tolerate salt, right, they're not going to be able to get cultured on this plate. They're going to be inhibited, right, by the salt. So therefore, this is a selective plate, okay, because it's selecting certain bacteria to grow on it, and then it's inhibiting others from growing on it. Now, it's also differential, okay, because it's going to help you differentiate based on the color change that you're going to see on the plate. It's going to differentiate, as I said, based on the color change, and basically those that are going to be able to ferment mannitol, they're going to change the color of the plate to a yellow color. So the side that turns, let's say you have two bacteria, okay? I mean, can you go ahead, by the way, split this plate in half, grow one bacteria on this side, grow another bacteria on this side? Yes. Now, if the plate changes yellow in color, that means that your bacteria can ferment mannitol, can ferment that sugar mannitol. If the pink agar stays pink as is, well, it means that your bacteria is not capable of fermenting mannitol. If you get any sort of growth, by the way, whether it turned yellow or pink, any sort of growth on a mannitol salt agar, that means that your bacteria is a halophile. Okay, it does love salt. Now, the pathogen Staphylococcus aureus, okay. By the way, how would Staphylococcus look like under the microscope? They would look clustered, right? Cocci clustered cells. Now, Staphylococcus aureus is a pathogen, okay? As I said, is it normal to kind of find Staphylococcus aureus in someone's nose? Yes, it is. On skin, yes. But the problem is if Staphylococcus aureus enters deep into the tissues, right, or in the bloodstream, it is definitely, going to harm the host, okay, and it can actually lead to death, and that's why it is considered a pathogen. So Staphylococcus aureus, that pathogenic kind, is halophilic, meaning it is salt-loving. And so it is going to be able to grow on a mannitol salt agar. And not just that, they're not just halophiles, but they're also mannitol fermenters. So if you have Staphylococcus aureus cultured on a mannitol salt agar, they're going to turn the plate yellow. Okay, so that's not a good indication, because if the plate turns yellow, then this tells you that you might be having, a pathogenic bacteria such as Staphylococcus aureus, cultured on it. Now, of course, if you're working with an unknown and your mannitol salt agar turns yellow, now you can't be 100% sure that it would be Staphylococcus aureus, but that's a possibility. Okay, you have to do, of course, more tests, but it is a possibility that it is Staphylococcus aureus. Now, the blood agar plate is another test. It's also differential because it's going to help you differentiate between the bacteria that are capable of breaking down red blood cells versus those that can't. But it's not selective. Okay, so it's not selective. Now, you can actually get 3 results, okay? This is the symbol for gamma, by the way, okay? So there is something called gamma hemolysis, where the bacteria growing on the blood agar will not be able to break down red blood cells. They're still going to be able to grow, it's just they're not capable of breaking down the red blood cells, so we call this gamma hemolysis. There's also partial hemolysis, and this one is called alpha, and this is the symbol for alpha. And so alpha hemolysis just means the bacteria are capable of breaking down red blood cells, but they're breaking it down partially, okay? How are we going to know whether it's an alpha hemolysis or gamma? Alpha is going to look murky green. The plate is actually going to start looking murky green. Now, one pathogen, Streptococcus pneumonia, the one responsible for causing pneumonia, is actually, alpha hemolytic. It's going to partially break down the red blood cells, and it's going to turn the plate murky green. Okay. Now there is another test or another result. Sorry. So let me kind of show you that one. And it's where the red blood cells are going to be completely broken down, and we refer to that as beta hemolysis. Okay, so this is kind of the symbol for beta. So beta hemolysis is complete breakdown of the red blood cells, and you're going to get like a golden appearance on the plate. I'm going to show you images in just a minute. Now, Streptococcus pyogenes, which is another kind of pathogenic streptococcus, which is the one that kind of causes strep throat. It's beta hemolytic. Okay. So see how the blood agar plate can actually, help you differentiate. Okay, that's why it's called a differential plate. So let's take a look at the results together. So this one right here, okay, is the beta hemolysis. Remember, what is beta hemolysis? Beta hemolysis is complete breakdown of the red blood cells. Here is the bacteria, okay, growing right here. Now, do you see how you don't see the red color anymore of the red blood cells? It's completely broken down. And you can even see how this person is holding the plate. You can even see their finger on the other side of the plate, because now the plate is like translucent, right? So the light is actually just like passing right through it. And this is an example of a beta hemolysis, complete breakdown. Alpha is a partial breakdown. And when you get partial breakdown, see how it's not very, it's still not very translucent, okay? So it's partial and it's kind of giving you that murky green color that I was talking about. Notice how this one, you could totally see the red blood cells. I mean, you could see the red color of the plate really. So this one is gamma hemolysis, which means no breakdown of the red blood cells is taking place. But the bacteria is still growing. And you can see the bacteria right here, okay? It just can't break down the red blood cells. And so that's why the blood agar plate is not a selective plate, but it is a differential plate. Now there's another test called the starch hydrolysis test. And so you start out with a plate. You can start out with, as I said, a TSA plate. But the thing is the media. Okay, so you go ahead and add starch to the media. So you have to make, gotta make sure that you've added some starch in there. Otherwise, the test is not gonna work out. And then, by the way, you can split your plate in half so that you can kind of, save time and save plates and media. So you can go ahead, split it in half, and you can actually inoculate one bacteria on each side. And usually with the starch hydrolysis, this is called, by the way, a starch hydrolysis test. We don't inoculate our bacteria kind of like in a zigzag pattern. We actually just do it in a straight line. Okay, so you go ahead, inoculate in a straight line the bacteria. And then you go ahead, after inoculation, you go ahead and stick it in the incubator for a couple of days. And what this test is, what is this test for? We want to know whether the enzyme can actually produce an enzyme called amylase. What is amylase? Amylase is an enzyme that can actually help break down starch. We actually produce it in our saliva, okay? So if you've ever taken a piece of bread and actually start chewing it, the amylase in your saliva is actually going to start breaking down the starch into sugar. So all of a sudden the piece of bread is going to taste sweet. So it turns out there are some bacteria out there that can also, kind of like us, produce amylase enzyme, and then some of them can't. So this test is going to help us figure out whether amylase is being released. What is amylase breakdown? Again, amylase breaks down starch. Okay. Now, how do we go ahead and figure it out? Well, after you've incubated your plate, take out your plate, and after incubation, okay guys, after incubation, go ahead and add iodine. Now, why are we adding iodine? Well, it turns out that if there is still any starch left on the plate and you've added iodine, it's going to turn black. Now, if it turns black, then that means it's a negative result. It means that starch was not broken down by the bacteria, and that's why it's still there. So if the starch is still there, okay, which means the bacteria was not able to use it, add the iodine and it will turn black. Now, if it doesn't turn black and you can actually get like a clear zone around the bacteria growth, that means that the bacteria was capable of using the starch and breaking it down, and it means that your bacteria was capable of producing the amylase enzyme. But if no clear zones and the plate turns black, then kind of like a purplish black color, that means negative, and the bacteria is not producing amylase and it's not breaking down starch. The starch is still there. So here is kind of like, here's kind of a result to help you out. So let's say here is bacteria A, and on this side you've inoculated bacteria B. Okay. Notice how you do see the bacteria growth. So here's bacteria growth, and here is the bacteria growth. Why is the plate black? That's because iodine was added. After you've incubated your bacteria, take it out of the incubator and flood the whole entire plate with iodine. Okay. Now notice how here the plate on this side is still pretty dark around the bacteria. Well, that means that the starch was not broken down. Okay, and that means the starch is still there. Okay, that's why it's a negative for starch hydrolysis. Hydrolysis means the breakdown of starch, so it means the starch was not broken down. On this side, notice how there's this clear zone around the bacteria. That means the bacteria is producing amylase, breaking down the starch around it, and therefore you get this clear zone right here. Okay, so this is definitely a positive result, and on this side it's definitely a negative result for starch hydrolysis. Another test is called the catalase test, and this one is going to help us differentiate between our Streptococcus and our Staphylococcus species. Now, Staphylococcus, by the way, they are going to be catalase positive, and Streptococcus are going to be catalase negative. So what does that mean? It means that Streptococcus cannot produce the catalase enzyme, and it means Staphylococcus do produce the catalase enzyme. So that's what it means by Streptococcus negative and Staphylococcus positive. Now, catalase is an enzyme that's going to help break down hydrogen peroxide, H2O2, into water and oxygen gas. So what we usually do is you go ahead after you've inoculated your plate and you've incubated everything, take your plate out. This test, by the way, could be done on a plate or in a test tube. Take your plate out or your test tube and go ahead and add some hydrogen peroxide. Now, why are we adding hydrogen peroxide? Because we want to know whether the bacteria has the capability of breaking down hydrogen peroxide by producing their own catalase enzyme. If you see bubbles, that means that your bacteria is producing catalase. So let me show you an example. By the way, you can kind of take a sample of your bacteria and put it on a slide. Okay, instead of adding hydrogen peroxide to your whole entire plate, just take a sample of your bacteria. So you would take like a slide. Okay, you would go ahead, kind of put a sample of your bacteria on the slide, and then you would go ahead and add, okay, some hydrogen peroxide. And then just wait, okay, and see what happens. If it starts to bubble, as you can see right here, that means that catalase was produced and it was breaking down that hydrogen peroxide that you just added, and that's why it's bubbling, okay? If after adding hydrogen peroxide nothing happens, then it's catalase negative, meaning your bacteria is not producing catalase enzyme. Okay, or as I said, you could do it like on, in a test tube, just put some bacteria in here, add some hydrogen peroxide, okay, and again, if it bubbles, catalase positive, no bubbles, catalase negative. So one more time, Staphylococcus, they do produce the catalase enzyme, so you should expect bubbles. Streptococcus is catalase negative, so you shouldn't see any bubbles if you're testing Streptococcus. There are a few virtual labs, and they are embedded in the worksheet itself, by the way. And you will, again, see some slides right here. And you guys need to go ahead and figure out the results. So you have some nitrate reduction test results to interpret, as well as litmus, and you also have some results for the MSA, the mannitol salt agar plate, and some results for the blood agar plates to interpret as well. And we also have some starch hydrolysis test results to interpret as well.